Hello and good morning. We're going to get started. It's 10 a.m. Eastern Time. Start saying hello and thank you for joining us today on our third international webinar series of 2013. This session is regarding basics in eukaryotic cell culture. My name is Mackenzie Ferrone, and I will serve as your host for this webinar session. Over the next 4 minutes, Dr. Tom Schreiber will present information on the essentials of cell biology, freezing of cells, and of the optimal culture substrates. If you have any difficulties with your audio during the webinar, we would like to remind you to please ensure you are listening directly through your computer speakers and the volume is turned up to a reasonable level. If you have difficulties, please use your chat feature to message the host, and we will do our best to get the issue resolved for you. So please feel free to use the WebEx chat as a vehicle to ask questions. You can submit your question at any time, and we will make a sincere effort to respond to all before the end of the webinar. We're so happy to have one of our own speaking with you this morning about these topics. It's my pleasure to introduce Dr. Thomas Schreiber, who is leader in the development of innovative cell culture products with Greiner Bio 1. Dr. Schreiber studied biochemistry at the University of Tübingen, Germany, and at the University of Toronto in Canada. During his doctorate studies, he gained expert knowledge in cell biology and in the interaction of stem cells with their microenvironment. Postdoctoral fellow, he focused on applied research and development in the fields of proteomics and bioanalytics. Today, Dr. Schreiber is responsible for the development of innovative cell culture products. And now I'm going to turn it over to Thomas.
Thank you for the kind introduction, and welcome to today's webinar on basics in cell culture. Cell culture is one of the most important techniques in biomedical research and biotechnology. When human or animal cells are isolated from primary tissue, they can be maintained with proper conditions in culture. To fulfill this, all the conditions that apply in vivo, such as the extracellular matrix of the cells or neighboring cells, metabolism, blood circulation, homeostasis, immune system, or body temperature, have to be transferred to the condition in vitro. For 15 minutes, I will guide you through some essential concepts and information around the culture of cells. The talk is structured into the 5 main tasks: cell banks, culture, the passaging of cells, cryoconservation, and I would also talk about contaminations. And I'd like to start with ways to obtain cells. And cell lines can be obtained from both commercial companies and non-profit organizations. I'd like to highlight some of the most popular non-profit organizations. There are main cell banks that distribute and store cell lines. The GCCP in the US, the ECACC in UK, the DSMZ in Germany, and JCRB in Japan. All of these facilities provide support with the identification of cell lines, and they host other microorganisms as well as antibodies. Furthermore, I would like to highlight some of the more specialized cell banks. If you are working with hybridomas, or if you are looking for monoclonal antibodies, the Developmental Studies Hybridoma Bank at University of Iowa, a resource created by NIH, might have interesting options. Cells, including induced pluripotent stem cells and embryonic stem cells, can be accessed via the Coral Cell repositories in the US, the UK Stem Cell Bank, and the Y Cell Stem Cell Bank in the US again. Noteworthy is the International Stem Registry, whose mission is to provide a searchable, comprehensive database that includes published and validated unpublished information on all human embryonic and induced pluripotent stem cell lines. The goal is to offer current information on all known human pluripotent cell lines, including those approved by the National Institutes of Health for federal funding and those derived through other public or private funding resources. This tree will include cell lines from non-profit institutions, academic centers, research enterprises, stem cell banks, and industry based in the United States and abroad. Aside from the mentioned institutions, there are, of course, various commercial suppliers for primary and stem cells. Next, I would like to highlight important aspects around the culture of cells. Everything with cells relies on the fact that the life cycle of cells is determined by the cell cycle. Briefly, the cell cycle is partitioned in 5 phases and typically takes about 20 to 24 hours for full completion. Post-mitotic G1 phase, the cells primarily grow and synthesize proteins, DNA is being repaired, and any missing cell compartments are completed. DNA application takes place in the S phase, which is followed by the G2 phase. In G2, proteins are processed, cell-cell contacts are loosened, and the upcoming cell division is prepared by production of specific proteins. This is completed, the cell enters the M phase, in which mitosis takes place. And when cells enter G1 phase again. If cells are to divide any further, they enter the G0 phase after G1. In G0, the cell cycle is on hold. This is a characteristic of differentiated cells, such as nerve, liver, muscle, or hematopoietic cells, and irreversible. Many can influence the cell cycle, such as severe defects in proteins or DNA that can either lead to a hold at one of the 3 checkpoints or apoptosis. Cell contact can guide cells into the G0 phase, whereas plenty of nutrients could trigger mitosis and thus act as a mitotic stimulus. On the other hand, lack of nutrients, such as by serum withdrawal, brings cells into G0 phase. This could be used experimentally, for example, to synchronize the cells. In general, cultured cells should be kept at exponential growth, which is indicated by a linear slope on the growth curve. Description of the cell number at different time points is very useful to determine the growth properties of cells. The steepness of the slope represents the population doubling time of a culture. The plateau of the curve indicates a confluent layer of cells. At this cells are confluent, undergo contact inhibition, and escape from the cell cycle into G0 phase. Primary cells are isolated from a tissue and cultured. They will proliferate for about 50 population doublings, then stop to divide. This is not the Hayflick limit and is caused by the progressive shortening of the telomeres with each mitosis. Hayflick and Paul Moorhead published in 1961 their finding that primary cells undergo several phases. Phase 1 is the primary culture that ends with the initial complete or confluent cell layer. Phase 2 is characterized by a high proliferation rate that slows down towards 40 population doublings. After approximately 50 population doublings, the proliferation ceases and cells undergo a crisis in phase 3, which is also called senescence, stop dividing ultimately, and undergo apoptosis. Cell culture, spontaneous changes through genetic instability can lead to a transformation with accompanying immortalization of these cells. The same mortality is an inherent feature of primary tumor cells and stem cells. In these cells, the enzyme telomerase is constitutively active and keeps the telomeres at a constant length. What is the fate of cells? What makes them grow? Factors can influence the cell culture, and the most obvious one is the cell itself. Cells derived from blood cells do certainly need different culture conditions than cells derived from colon, for example. Also, age and patch number can determine the cell's morphology and can cause differences in cell cycle or metabolic functions. On the basis of morphology, that is, the shape or the appearance of cells, or on their functional characteristics, cells can be divided into 3 types. Epithelial cells are derived from one of the 3 layers: endoderm, mesoderm, or ectoderm. They are involved in the functions of organs such as gas exchange in the lung, directed transport in kidney, and gut, or the barrier protection in skin. Epithelium can consist of 1 or more cell layers, and the cells are oftentimes polarized. have a polygonal shape, and their appearance can vary from non-confluent island-like structures, shown here for the Keiko cells. This is a colon comma cell line, to more or less equal growth, such as with the HeLa cells, which are derived from a cervix carcinoma. Cells of the connective tissue, such as fibroblasts, appear more elongated as epithelial cells. Note the different morphology of mouse embryonic fibroblasts, NIH3T3, compared to the CACO2 or the HeLa cells. Retention cells do not attach to the surface of the cell culture disposable. They grow mostly free-floating and do not show contact inhibition. These cells can be derived either as primary cells from tissue or tumor. Alternatively, established immortalized cell lines can grow almost indefinitely. The fact that the cells can grow endlessly doesn't necessarily mean that one doesn't have to care about passage numbers. In fact, there are various reports highlighting the differences regarding cellular functions in between different passage numbers. Shown here are examples of cellular functions such as polation or permeability affected by passage number of Keiko2 cells. So it is best to perform assays with early passage numbers, as these resemble best the original cell. Caution is advised when comparing experimental results from very different passage numbers. Therefore, it's actually best with a pyramid to stick to the same passage range. The authenticity of cells in use should always be a matter of concern. A prominent example is the contamination of various cell lines with HeLa cells, for example. You can see from this table there are also other cell lines that have been reported to be contaminated. The confirmation of the cell line identity should include methods such as karyotyping, fingerprinting, either enzyme profiling. And this should be done regularly in the cell culture lab. We will see which physiological parameters contribute towards a controlled environment for the cells. All biological processes are temperature dependent, and for eukaryotic cells, 37 degrees Celsius is the optimum temperature with regard to their enzymes. Cells are usually grown in a fully humidified atmosphere in the incubator to avoid any detrimental evaporation effects. That is required for oxidative metabolism can influence cell cultures as well. The oxygen transfer rate is determined by the area and the path length that is available for oxygen diffusion. For such disposables, cells should not be covered with more than approximately 5 millimeters of medium. Besides the temperature optimum, there's also pH optimum for cellular processes, and this is adjusted with buffers in the medium. Addition of sodium hydrogen carbonate to the medium allows for an open buffer system that relies on CO2 in the incubator. CO2 carbon dioxide reacts in the medium with water to hydrogen carbonate and a proton. The diagram illustrates the dependency of sodium hydrogen carbonate in the medium from CO2 in the incubator. To achieve a physiological pH of 7.4, almost 10% CO2 are required for DMEM medium, which is represented here and features 3.7 grams per liter of bicarbonate in the medium, in contrast to the 5% CO2 that are required for the minimum essential medium to achieve a pH of 7.4. And this is due to the higher amount of bicarbonate in the DMEM medium. Alternatively, heat can be used in the medium as a buffer, allowing for a CO2-independent cell culture. Note that heat buffers should be stored strictly in the dark due to the phototoxic activity of heat. A potential change in pH followed by the addition of pH indicators such as phenol red to the medium. Cell growth rate would eventually lead to a more yellow color of the medium because of the accumulation of protons due to the cell metabolism. In contrast, the very low ambient air levels of CO2 lead to an alkalization of the medium with time. While it might be alluring to feed a new cell line with medium that's just around in the lab, another choice is to stick to the recommendations of the aforementioned cell culture banks. By typing the name of the cell line into the search window of the cell bank website, all relevant information appears within seconds. They are not only on the media, but also on doubling time, subcutting ratios, and general background information. Some very common cell culture media are shown in this table. Besides the modified Eagles media, MEM and DMEM, I1640 and HEMS F12 are very common and universal media. Hoy's 5A and Leibowitz L15 are media used for primary cells. Note that the DMEM should be used with 10% CO2 and L15 with 0% CO2 actually. Free applications, a mixture of DMEM and HEMS F12 has proved to be beneficial for the cells. The same combination in a formulation without calcium and magnesium works well for suspension cells. Nutrient consumption can be assessed from the media by the pH, or by measuring the pH, the glucose decline, or the appearance of cell debris. Cell debris indicates that actually the medium is not in a very good shape anymore. Probably the most important medium component is serum. It's applied from 1 to 25% to the medium, depending on the cell type. The most used serum is fetal calf serum. It contains a wealth of mitogenic factors, nutrients, and adhesion proteins, adds essential fatty acids to the medium, as well as buffering capacity and protease inhibitors. However, due to its animal origin, serum is a potential source of contaminants and relies on batch-to-batch variations. Data requires a batch testing that all commercial suppliers of serum offer. Most protein solutions, repeated freeze-thaw cycles are not recommended. The complexity of its mixture makes it sometimes not preferable for certain experimental setups, such as migration assays. The downside of omitting serum might be decreased adhesion and proliferation of the cells. In many labs, heat inactivation for 30 minutes at 56 degrees Celsius is performed to inactivate potential contaminants and complement components. However, with the modern filtration methods applied by the manufacturers and the quality standards, it's unlikely that contaminants such as Mycoplasma remain in the serum. For certain immunological questions, cytotoxicity assays or virus screening, complement inactivation might be useful, but for the majority of applications, the detrimental effects of heat inactivation towards the actual beneficial components in serum make heat inactivation most likely undesirable. Here, supplements include amino acids, various salts and buffers, maybe serum replacements, and antibiotics. When culturing epithelial cells, it might be beneficial to use amino acids that are actually the D amino acids, because fibroblasts would not be able to metabolize the D amino acids. They can only metabolize the L amino amino acids. Glutamine is a very important nitrogen source for the cells and is usually supplied with psyllium. However, meanwhile, it's very common that glutamine dipeptides are added instead of the glutamine because they are more stable than the actual glutamine. There are several serum replacements available. So if one wants to stick to a more precise or better formulated composition, then these serum replacements are quite a choice. With regard to the antibiotics, it's still very common to use antibiotics on a regular basis in the medium. However, this is not recommended because it might mask any contaminations that appear. And therefore, antibiotics should only be used in an acute phase of a contamination, or when cells are isolated and cultured for the first 2 or 3 weeks. Now I'd like to talk a bit about the culture substrates that can be applied on the cell culture vessel. This is a chart resembling or showing how the cell surface looks like. Within the membrane, there are the integrins, which are calcium-dependent proteins that can actually link to extracellular matrix molecules. There are, as an extracellular matrix component, collagen fibers at the outside of the cells surrounding the cells. There are proteoglycans and polysaccharides at the cell membrane. And all these might interact with a potential surface on which this has cultured. On the other, our standard tissue culture surface consists of polystyrene. Natural virgin polystyrene that is untreated is rather hydrophobic. That means if you would apply drops to a polystyrene vessel, they would appear not flattened instead of like round shaped. And for this reason, polystyrene needs to be treated by a physical treatment to use functional groups such as containing oxygen or nitrogen to the polystyrene, which actually then makes the polystyrene hydrophilic and also allows the cells to attach to this charged surface in a better way. Adherents can interact with their integrin receptors, for example, or with their proteoglycans with any cell culture vessels. And the first stage of cell attachment would actually be that the functional groups of the polymer would interfere with the proteoglycans, which is an electrostatic attraction then, followed by interaction of the integrins with serum proteins that cover the polystyrene vessel. Fibronectin, for example, is a very common, one of the most represented serum proteins, and is also known to be as a very standard protein for the cells. Lastly, cells would secrete their own extracellular matrix proteins with which integrins on the surface then would actually interact. Depending on the need the original cells originate from, several extracellular matrix proteins that are available from commercial suppliers can be used to coat the polystyrene vessel in order to facilitate the cell adhesion to the polystyrene surface. Very common are collagen proteins, fibronectin or laminin, ECM gels such as Madrigel can be used especially for the culture of stem cells. Fibronectin is a very good substrate for stem cells as well as for endothelial cells, melanoma and osteosarcoma, live cells. And tenacin might also be a substrate, although not a very common one. So if you experience problems with the adhesion of cells, I would recommend using collagen or fibronectin as a starting point to improve your cell culture. Grind1 also offers several surfaces for cell culture. We have a range of non-biological surfaces that are, of course, animal-free. These are sterile products that feature a rather low shelf life. They can be stored at room temperature and include our standard CellStar tissue culture treated products. We have a advanced TC surface that is a polymer modification and can be used especially for sensitive cells or primary cells, as well if you would have a lot of washing steps that could drive the cells off the surface included in your assay or experiment. We have a standard suspension surface that is very good for suspension cells in general, and we also have a special surface called Serum-free that is a low attachment surface which is very good for the kind of spheroids or embryoid bodies, for example. From these non-biological surfaces, we also have a range of coated, pre-coated products, so you could actually buy them and you are ready to go. You don't need to do any coating by yourself anymore. We have poly-D, poly-L-lysine coated plates. Poly-D and L-lysine add a positive charge to the cell surface, which makes it easy for the cells with their merely negative proteoglycans to interact with this surface. This is kind of a standard biological coating that works well with a variety of cells. Modularized are our biological coatings, collagen, fibronectin, and laminin. All of these are applied in an aseptic way. Collagen features a shelf life of 2 years, whereas fibronectin and laminin a shelf life of 6 months. Most of these surfaces can be used with primary cells, depending on the cell type. Collagen 1 is especially good for bone cells or mesenchymal cells. Fibronectin works very well with a variety of muscle cells or epithelial cells, where laminin is especially suited for neuronal cell differentiation or the culture of neuronal cells. I will give you an example why the surface actually matters for the culture of cells. One common experimental conditions for human embryonic kidney cells, AK293, is to cultivate in a serum-free medium, and this certainly adds some stress to the medium, to the cells, because the serum would actually add fibronectin, for example, or other adhesion factors which would be lacking in the serum culture. And this is easily can be imagined by, as you can see here, the initial adhesion of the cells with a standard CellStar TC treatment is not very well to our advanced TC surface or to the PDL coating actually, and also the long-term adhesion or proliferation of the cells shows that for a serum-free cultivation of cells, actually the polymer modification advanced TC or CELLCOAT PDL as a biological surface are better choices for the cells. Subcutting of suspended cells is fairly easy and can be done by aliquoting the required cell number to fresh medium. Alternatively, cells can be spun to the bottom of a tube. The cooked medium then can be removed or replaced by fresh medium. Adherent cells, on the other hand, can be subcutted either by shaking or scraping of the cells with a rubber policeman or by applying an enzyme. Shaking of the cells is a very gentle method, but it can only be applied to loose adherent cells. Screen the cells off from the substrate with a cell scraper of the digestion of proteins on the cell surface, but can damage cells and hardly gives a single cell suspension. The enzymes are the best choice for standard subculturing of cell lines. It's very important to apply any enzyme solution to the cells only as long as it is really needed to not damage the cells. The cause of cell attachment can be easily followed with a microscope. On this slide, you see an almost confluent layer of HeLa cells. Removal of culture medium, followed by one step with phosphate buffered saline, trypsin was added. Once after applying trypsin, most of the cells detached and appear rounded. Some cells would still attach to the tissue culture dish. 5 minutes after applying the trypsin, all cells are in suspension, and trypsin should be inactivated by the addition of at least 3 volumes serum-containing medium compared to 1 volume of trypsin. This is the most common detachment enzyme as it is very efficient and comes as a ready-to-use solution. It is both as a purified and a recombinant enzyme, and there are combinations of trypsin with EDTA available. EDTA chelates divalent ions. At higher concentrations, a drawback of trypsin is the cleavage of cell adhesion molecules that might be subject to further investigations. In this case, acutase, for example, might be a better option as it is more gentle to cell surface proteins. For special requirements, such as samples rich in connective tissue with elastic fibers or single cell suspensions are strictly required, allelase and pronase might offer interesting options for the investigator. In general, these enzymes are very strong and thus could potentially harm the cells. Enzymes such as hyaluronidase, disbase, or collagenase are less toxic than trypsin, but at the expense of incomplete dissociation of the cells. However, this can also be turned into an advantage, for example, for the detachment of whole cell sheets. Hyaluronidase cleaves hyaluronic acid and thus facilitates permeability of tissues. It can be used to remove cumulus cells as well from the primary culture of mouse embryos. Especially useful in avoiding cell aggregation of cell suspensions due to DNA release is DNase. For all enzymes covered, it is important, of course, to apply the temperature and pH conditions required by the respective enzyme. For detachment, PBS without calcium and magnesium should be used because these enzymes are required for integrin-dependent cell adhesion, as I mentioned earlier. And I also mentioned earlier that the respective enzyme should really only be applied as long as possible or as long as necessary. This is easily followed with the microscope. Most enzymes have to be diluted or removed from the cells, where trypsin is inactivated through the addition of serum-containing medium, which also serum contains a trypsin inhibitor, and in this way trypsin will be inactivated by serum. If cells are very hard to detach, which can happen especially with primary cells, washing the cells with EDTA-containing PBS, the addition of collagenase to the enzyme of choice, or gentle tapping of the vessel might also help. Covering the relevant aspects from obtaining cells, culturing cells, and detachment of cells, it's time to have a look now at some backup strategies for cells. The cells are oftentimes required only for certain aspects of a project or experiment. It makes sense to freeze cells regularly. Earlier in this talk, I mentioned that cells are kept in culture for a long time, are prone to undergo a genetic drift or senescence. This makes it preferable to store as many cells as possible at early passages in special cryo vials. If contaminations would occur, for example, potentially contaminated cells could be discarded and cells from stock could replace them. The freezing cells is the fact that most cellular reactions freeze at the glass transition temperature of water at minus 137 degrees Celsius. There's not enough energy available for metabolic processes, and there are also no ice crystals due to the absence of liquid water at that temperature. This temperature can be achieved in the gas phase of liquid nitrogen. We recommend the storage of cryo vials in the gas phase instead of in liquid nitrogen itself to avoid potential Mycoplasma contaminations and the potential entrance of liquid nitrogen into the vials. The latter can lead to bursting of the tube with improper handling. Therefore, it's very important to follow the safety measures related to the work with liquid nitrogen. To avoid the formation of ice crystals within the cells that could significantly damage the cells, storage of cells for longer than overnight at minus 80 degrees Celsius must strictly be avoided. The common cryoprotectants in cell culture medium, besides a higher amount of serum, are dimethyl sulfoxide and glycerol. Both leave their protective effect after penetration into cells and prevent the osmotic dehydration of cells as well as the formation of large ice crystals. It passes the membrane readily but is toxic at room temperature. Glycerol, on the other side, is less toxic but cannot be removed as easily from cells as DMSO. Therefore, protocols for these two cryoprotectants are not interchangeable. When freezing cells, it's best to freeze cells at a low passage number and to avoid cell clumps. Of course, cells should not suffer from contaminations and have a high vitality or viability, be achieved by freezing them at about 8% confluency. It's always advisable to keep a culture running until an aliquot of frozen cells had been thawed again after 1 to 2 weeks for cell viability. Detailed freezing protocol is available from the technical appendix in our catalog or from our website. A very important issue with regard to sample backup is the identification of cryo vials. Small sample archives, filling of vials by hand is sufficient, though prone to errors such as fading ink, for example, or indecipherable handwriting. Coded tubes, on the other side, can be ordered custom-made and are clearly a better choice because both linear and data matrix codes are machine-readable, and with data matrix code accepting up to 25% of the code area being unreadable. In a cryo vials with data matrix code can also be placed in a rack that allows for bulk reading. As a last part in my talk today, I would like to spend a few minutes with the diagnosis and removal of contaminations. Probably everybody working in a cell culture lab faced this scenario at least once. After opening the incubator in the morning, the phenol red indicator in the cell culture medium had turned yellow, turbid, and cells are either dead or severely affected by a contamination. Most likely the source is bacterial contamination due to improper aseptic technique in the cell culture lab. Many bacteria can be recognized fairly easily by the massive propagation and their movement that is visible at high magnification in the microscope. Their propagation exhausts most of the nutrients in the medium. The diagnosis can be assured by plating the medium on LB agar plates, followed by a microbiological examination. Fungi become apparent in the cell culture by the appearance of hyphens, as you can see in the left picture here. Besides cells, laboratory equipment such as the water bath can be afflicted with the appearance of fungal flakes. Fungus causes the medium to turn basic in contrast to the acidic medium that applies with bacterial contaminations. Yeast, which is a subclass of fungus, can be identified by the occurrence of small groups of round or oval yeast cells, such as shown in the picture on the right by the white structures. To the appearance of fungi, it's very important to keep the water bath in cell culture labs as clean as possible. This can be done by general regulations such as wearing gloves, keeping doors and windows closed, of course regularly changing the water, and also by the use of a water preservative. Mycoplasma are small prokaryotes that vary in size between 0.2 and 2 micrometers. Thus, they can pass common sterile filters. They grow on the surface of cells, and approximately 30% of all cell lines are estimated to be contaminated. They can impact the cell metabolism, and since they are small in size, they are not as obviously detected as bacteria or fungi. Mycoplasma contaminated cell lines can be identified by DAPI staining. DAPI is a DNA staining fluorochrome that is usually used to stain cell nuclei, shown here on the right, here. And the stain pattern on the left is a hint for Mycoplasma contamination. So instead of the DAPI staining only appearing with the nuclei, you'd also see it like in the it would appear as it would stain the cytoplasm. The best way to detect Mycoplasma contaminations is with a PCR, as this gives also definite information on the source of the Mycoplasma. With CytoCheck, Griner Bio1 also offers a convenient kit for this purpose. If you experience contaminations in cell culture, it's best if the contaminated cell culture if the contaminated cell line is discarded to prevent the emergence of resistances and also to prevent any escalation of the contamination in the lab. However, for some reasons, under some circumstances, this might not be possible or not wanted, and then a variety of antibiotics can be used actually. You see a range of antibiotics, and one should note that the working concentrations and the stability of the respective antibiotic under culture conditions, especially at 37 degrees. This means that the antibiotic has to be replaced mostly every phase, basically. The regular use of antibiotics in cell culture should be avoided because this can mask any potential contaminations. So once primary cells are isolated or once contamination is gone and also has been approved to be gone, then the antibiotic should actually be left out of the medium. Effects with regard to contaminations or any weird observances in cell culture are virus infections. These are not very common in cell culture in contrast to cell contaminations that were mentioned earlier in this presentation. A rather interesting cell growth pattern is obtained when vibrations interfere with the cell culture. Depending on the wavelength of the vibration wave, cells grow in circles more or less apart from each other. In case of such a phenomenon, careful investigation of the incubator environment has to be performed. For example, for the presence of the incubator or any other moving equipment. Lastly, at the end of my talk, I would like to mention some literature resources that are very useful. There are three main literature lines. These are current protocols, methods in molecular biology, and nature protocols that give hands-on expertise and very detailed protocols on basically every aspect of cell culture. So it's very useful resources if one is searching for a protocol, for example. Furthermore, there are guidance books that give some more general advice on common issues. Two of them in English language and two of them in German. And of course, there are the standard theoretical books such as molecular biology of the cell or molecular cell biology or the cell that give the necessary background advice. I will conclude with the main concept of good culture practice, which is actually according to good laboratory practice. Kind of standardizes the cell culture. Of course, the scientists working in a culture lab should stick to the GLP, to the good laboratory practice. Some other issues are that you should actually adequate your medium because one, you wouldn't bring contamination into the whole medium bottle, and second, you should also warm up medium only as aliquots because if medium is not used by amount, then the frequent warming might lead to stability, for example, of glutamine, and therefore actually it's best to aliquot the medium. You should handle only one cell line at once in the cell culture hood. And the problem of cell cross-contaminations, and I would assume that this is one of the reasons how these occur if people handle two cell lines at once in a cell culture cabinet. You should definitely know your cells or your cell lines, which means that you should check for their identity regularly to make sure that you wouldn't see any adverse effects or that your assay really shows effects that depend on your cell line that you want to use. I mentioned it earlier, you should avoid the regular use of antibiotics. Using antibiotics is not an excuse for having an aseptic technique in the lab and would also only mask contaminations. You should test your cells regularly, for example, with karyotyping or isoenzyme profiling. A general advice is to have a special cell culture code and, of course, gloves in the cell culture lab. It's actually not advisable to mix cell culture codes with regular lab codes. Cell culture lab should have a special cell culture code. You have a good subculturing routine, meaning cells should basically stay in the exponent phase or in the log phase of their growth. They don't become too confluent because this can lead to changes of cells. Very important to freeze aliquots, especially if you are generating stably transfected cell lines or if cells have a special feature. And also it's important to freeze the aliquots at an early passage so that the cells would most likely still have their original phenotype. Last but not least, Of course, a clean equipment is very important as well. So cell cabinets should be cleaned regularly, and also the incubators should be cleaned regularly. This I'm coming near the end of my talk. I would just want to highlight some of the support Griner by One can give to you. We have an FAQ database on our website, which handles questions that we receive on basically all of our products, not only the culture products. We have a wide range of application notes, and what we call forum is a review article to a certain topic or issue. And we have a webinar series that are going to be good in this year. With I would like to thank you for your attention, and I hand over to Mackenzie again.
Great. Thanks so much, Thomas. Before we end the session and answer some of your questions, I want to take a minute just to thank everyone for attending our webinar series today, and to thank Dr. Schreiber for such a wonderful presentation. I would also like to remind everyone that our webinar series will be continuing throughout the rest of the year. In October, we will be talking about 3D cell culture. We'll get information and registration options out to you. We'd like to let everybody know that recordings of this and all webinars will be available to those who would like them on our U.S. website, and we will also be emailing them out to all attendees after this webinar. We'll get that done as quickly as possible. And if you would like any information on the literature pieces that Thomas has shown you, or product samples dealing with anything that you've heard today, please email us. I have put my email address in the chat box for everyone to see. Feel free to email me with any other questions or concerns or needs that you might have. Now we're going to move on. We do have some attendee questions for you here, Thomas. I'll start off with a person asking, sometimes when they culture some cells, they can see that some small black dots appear around the cells, but it looks like bacteria since it's much bigger than bacteria. The dot can't move, but it can grow more and more during the cell culture process. Do you have any idea what this dot could be?
I mean, from my position now, it's somewhat hard to tell, of course. I would think of maybe just cell debris that would accumulate during the culture. Another possibility, the cell line you're using, any vesicles leaving like the cells, or small like vesicles derived from membranes. As I said, it's very hard to tell without actually seeing. But feel free to email me a picture, and I would be happy to have a look at it and get back to you on that issue.
Okay. Another question from an attendee. In what instance would not want their cells to adhere to the culture surface and form fibroblasts? Currently, they are working with a line of human embryonic kidney cells, and the way they determine their health is by confluency and fibroblast formation.
What you're doing is right, I guess. There might be some reasons. If you're working with CHO cells and you want to culture them more for biotechnological purposes, then you would maybe want them to become non-adherent. Furthermore, if you're working with stem cells, it might be applicable that you grow them as embryo bodies or as spheroids. In these cases, non-adherent cell culture is good, but actually, if you're working with blasts, then I think adherent culture is fine.
Okay. And we've gotten a few questions around this topic. If you could tell people what you use to clean your incubator, or what process do you use to clean your incubator and culture hood? A specific question is 70% ethanol sufficient to do this cleaning?
Well, basically 70% ethanol is completely right. There's no way of going 100% ethanol, because this actually preserves any spores or bacteria. there has to be a certain amount of water in the ethanol. So 70% is totally fine. That's actually a perfect reagent to clean your such a cabinet. Some people like to use Mycoplasma removal agents. I used them during my lab career also in one lab because it was very common over there. But honestly, I do not know whether they are really efficient. I cannot, I do not know of any reports that them really adds to purity in your cell culture lab. So yes, 70% alcohol is fine. Ethanol is fine.
Right. For cryopreservation, do you prefer vials with the threads on the outside of the tube or the internal threads? Because this person has vials explode during warming, and they've heard that the internal threads might fix this problem.
Actually, it doesn't matter which type you use. Some people like external thread, some internal thread. I think the likelihood that you would contaminate thing is maybe lower with an external thread, because you could actually basically touch everything on the outside, whereas with the internal thread you have to make sure that you only touch really the grip or the stop part of the thread. So basically it really doesn't matter, and it's always advisable to fill the cryo vial not to the limit, so you should stick to the graduation that your manufacturer actually gives with the cryo vial.
Okay. And can you go over briefly the pros and cons of filtering media after the addition of supplements?
Mm-hmm. One, I would like to stick to the last question. One important point is that we recommend only the storage of the cryo vials in the gas phase of liquid nitrogen. And to my knowledge, this is what all manufacturers actually recommend, because this decreases the likelihood of liquid nitrogen entering the tube. So maybe this was also be part of the problem. So I apologize. Now the next question was pros and cons of filtering media. Personally, I've never filtered media. You should have the appropriate safety measures, and what should be filtered with the media, or if you use serum, mycoplasma would pass the filter, and the ingredients that you receive should also be sterile. So for sure it wouldn't do any harm, the filtering process, as long as it is done in sterile condition, but in my opinion it's not really necessary.
Okay, great. We have about 4 more questions here that people have brought in, and I think we're going to stop doing the live questions at that point, and any others will be answered via email. So I just wanted to let everyone know, if you have another question, I will record it and we'll get it answered. We just want to stick to the 1 hour mark. So I've got 4 questions for you, Thomas.
Okay, great.
When switching media types on primary cultures, do you recommend a 50-50 split first, or is it okay to go straight to the new media? Will that have any effect or change on the stress of the cells?
I would definitely go 50-50 first. So, for example, if we go serum-free, but also if you change medium, then I would do it gradually. So I would do the first medium change with 50-50, then keep the cells for 2 days or 3 days. Have a look at them. You likely know how your cells look like when they are happy, and if they go well. So stick 50-50 until they look fine, at least for 2 days, until you passage them for the next time. If we do the serum-free, then we go to 25-75, and then we do even another step at half and 87%. So yes, you should really do it quickly, because cells to their medium, and if you change it, this could add stress depending on from what medium you apply, and doing it readily would definitely lower the stress for the cells.
Okay. And if you're treating a culture for contamination and an antibiotic doesn't seem to be working quickly, would you recommend trying another antibiotic?
Definitely. I would definitely try another one. I've had, this is when the standard penicillin streptomycin just didn't work, and then after another cure with gentamicin, the contamination was gone. Also, you have to be aware what the specificity of your antibiotic is. Some antibiotics only go for gram-positive or gram-negative bacteria, if it is a bacterial contamination. So you also have to be aware of what the antibiotic actually targets.
Okay. And for hybridoma cell cultures, do you recommend different media for cell maintenance cultures versus cultures in bioreactors for antibody production?
It depends on what kind of bioreactor you're using. If you are using some sort of stirred bioreactor, this might add an increasing amount of stress to the cells. Therefore, there are like media supplements such as pluronic. These lower physical stress caused by the stirrer to the cells. Also, you might actually have special requirements with regard to the production in the bioreactor. Basically, it depends a little on the bioreactor you're using. If the cells are happy in the bioreactor, then obviously you don't need to change the medium. However, it could be the case that you needed to add something to lower the stress due to the stirring.
Okay. Last few here. Is there a difference between 70% ethanol and 70% isoprene as far as effectiveness for cleaning surfaces?
Basically, no. I've read reports that say that isoprene might even be a bit more effective with regard to ethanol, but actually it doesn't matter. Ethanol would be just fine because it's used like in hundreds of millions of labs worldwide, and so you just go with that.
Okay. And then one last question. How long can frozen cells be stored at negative 80 degrees Celsius?
I'd say a very, very long time, because from hematopoietic stem cells that were frozen, there's a study going on hematopoietic stem cell freezing process, and how they would behave after they are thawed again. And now I guess they are at the age of 25 years, so definitely cells could be stored in liquid nitrogen for an unlimited time. But the question, I guess, was about minus 80 degrees. Is that right, Mackenzie?
Mm-hmm.
80 is only overnight, definitely. As I mentioned before, that at minus 80 there could be still ice crystal appearance or building, or ice crystals showing up in the cell, which definitely would disturb cells. So what I would suggest is, our protocol actually is, we add the cryoprotectant medium to the cells, let it stand for about 10 minutes so that the cryoprotectant can enter the cells. Then we go for 2 hours at minus 20, and then after that we go overnight at minus 80, and after that into the liquid nitrogen tank.
Okay, great. Well, thanks so much, Thomas. I believe that's all the questions from the attendees we have. So those of you who are still on the line, we want to thank you again for your participation. We love having the questions, and we love having all the attendees. When you exit out of this webinar, you will be automatically directed to a very short survey that just helps us understand how you enjoyed your experience today, how might improve further in the future. So we would really appreciate it if you took 2 or 3 minutes to fill that out for us. Again, you will be getting an email from us with a recording of this presentation in case you want to refer back to it in the future. And with that, we thank you again, Thomas, and all the attendees, and we hope that everyone has a wonderful day.